at each and every little cube of the brain, because you divide it up into little cubes, they're called voxels, how the response in that part of the brain matches your statistical model, which then gives you this thing called a statistical parametric map. And these, everyone calls them the pretty pictures that neuroimagers get. All this is a map of the statistical significance of the fit of your model to the brain activities. You can see here, it's a highly significant fit here in the posterior area. So this is visual cortex. And here you can see the lateral geniculate nuclei. Because I told you this is just a checkerboard flashing up and down. And you then do a bit of statistical inference on that to see whether your hypothesis is upheld or not. So I'm going to talk to you about fMRI in the context of lots of different things. I believe in having quite a multimodal approach to studying memory processes. And at the end, I'll get down to some other techniques like MEG and something called transcranial magnetic stimulation. So just a simple functional MRI study. So this is quite an old study that we published in '99. So what people were doing while the scanner was on is they were learning a rule system pertaining to these 4-letter strings. And these 4-letter strings were shown and then repeated and then repeated. But there was an important change. That every so often the typeface in which the letters were presented was altered. So why did we do this? Basically, as I told you before, the hippocampal damage results in a dense anterograde amnesia, so an inability to learn new things. So the hypothesis is if the hippocampus is involved in processing novelty. So this, by changing the typeface in which these letters were presented, we introduced a type of perceptual novelty. And what we showed was that the anterior hippocampus here on the left shows a very focal activation. And as you can see, when the font is changed at this moment, the response increases and then it decreases with repetition of the font. So that's a perceptual novelty response. What we also did every so often was to change not the rules, but the actual letter strings. You see these are X's and Q's and A's, but then we just changed them to R's and H's. So all the rule system was the same, but this is a type of exemplar novelty which was behaviorally relevant. So then it had to map the rule they'd learned onto these new letter strings. And what we showed there was this is the critical thing, that the left anterior hippocampal response was present also. You can see here in the same part of the brain was also present when the exemplars changed. Now, much to my disbelief, we also saw an activation when they became more familiar with the exemplars. So anecdotally, so in the old days of this software, it's called SPM. It's developed in London, and anyone who works with functional imaging uses this software. In the old days, it used to test for the effect of interest, but then do the opposite test automatically. So I let this analysis run over the weekend, and it came in on Monday, and saw a big hippocampal activation bilaterally. But for the opposite effect, I wanted to see a novelty effect, but this showed a strong familiarity effect. But then, of course, I looked at the novelty effect, and it was just in a different part of the hippocampus. So it was anteriorly in the hippocampus, whereas the familiarity effect was posterior. So this sort of started a new idea that perhaps the hippocampus is quite a long structure, as I showed you in that first slide, and maybe the anterior part of the hippocampus, so the front part, is doing something different to the posterior part. And so the idea was that when new things are processed, it activates the anterior hippocampus, as you can see here, and when familiar things are processed, it activates the posterior part. So this was quite not very well taken by some of the old-fashioned people working on the hippocampus, but it does actually turn out about 10 years later. There's some agreement now in the literature that the dorsal and ventral in rats pertain to the posterior and anterior hippocampus, just the shape is slightly different. So they do seem to be quite functionally distinct structures. And it's quite important for some pathologies, because different pathologies affect different parts of the hippocampus along this long axis. So you can see from a very simple fMRI study that was showing these letter strings and changing the fonts and changing the letters, we picked out quite powerfully a new idea pertaining to one of the most mysterious structures in the brain. There's another part of the medial temporal lobe which is located just around the hippocampus. You may have heard of it. It's called the perirhinal cortex, and it's located just here. You can see this is the entorhinal cortex. You might have heard of it because that's the first thing that gets damaged in Alzheimer's. But just here, next to the entorhinal cortex, you have the perirhinal cortex. And this became neuroscience became very interested in this part of the brain in the early 90s, because it was shown that neurons in this part of the brain also had novelty-sensitive responses. And they also demonstrated that if you took out the perirhinal cortex in monkeys, you would get a type of amnesia similar to the hippocampal damage. And then some colleagues in Germany were doing some human intracranial recordings. So these is another powerful way to look at the brain function in humans. So you take advantage of the fact that when patients come for assessment of their epilepsy, these are epilepsy patients that have medial temporal epilepsy, they quite often have electrodes implanted into their brains to see where the epileptic activity is coming from. So the idea is to work out where the seizures are coming from, to then take out that part of the brain. These patients have had epilepsy all their lives. It's very serious. It doesn't respond to normal anti-epileptic drugs. So this is kind of a last resort. But what they were doing at this stage in Germany, and we started doing in the last 6 months here in Madrid, is also doing cognitive tasks while these patients are waiting with the electrodes implanted. And what this German group did is they did a memory test where they were looking at encoding-related activity in the hippocampus and the perirhinal cortex, and they showed, you can see here, that when a word is successfully encoded, you get a bigger response in this part of the brain. So this is what's called an intracranial event-related potential. So this is on a fast time scale. So this is 400 milliseconds. So this is peaking at around 400 milliseconds. So you see a response here in the perirhinal cortex that preceded a memory response in the hippocampus. So we wanted to do something similar. Until that stage, imaging the perirhinal cortex had been thought very difficult. It's quite anatomically variable, and it's quite a small structure. So we devised the scanning protocol that was very, very small resolution. So these voxels, what I call them, little cubes in the brain, were very small. We did the same task, just looking at this part of the brain. So this is the hippocampus, and you're looking at it side on. So focus our tests on the hippocampus. This is only to remove the effects of any eye movements. And we scanned them while they were learning words, and then had a distractor task, and then did a free recall. And we did something, this is called the subsequent memory effect, which is worth remembering. There's a lot of memory research uses this. Particularly, it was first done by people doing EEG recordings. So the subsequent memory analysis, what you do is you look at encoding, memory encoding during fMRI, and then you look at when you do, say, a recognition test afterwards, you then see which words were remembered and which were forgotten. And then you go back and say, okay, so this one was subsequently remembered and this one was subsequently forgotten. And you do a comparison on those. So it's the brain activity related to successful encoding. And what we showed, so this is looking at the brain but cut this way. So it's called the coronal section. And this blow up here, A is for the amygdala, which sits above the perirhinal cortex, which is here in the collateral sulcus. And you can see here that we did manage to image some perirhinal activity.